Hello students, welcome to EPG Pathshala. I am Dr. Seema Kalra from Indira Gandhi National Open University. In this module, we are going to talk about the fundamentals of gene therapy under the paper Molecular Therapeutics. Gene therapy is one of the direct applications of recombinant DNA technology, which has rapidly progressed in the last 3 decades due to its promising outcomes. It works on the idea of using genes rather than drugs for the treatment of diseases. Gene therapy has shown potential in treatment of various inherited genetic diseases like cystic fibrosis, muscular dystrophy, sickle cell anemia, as well as diseases like cancers and AIDS. But studies are still in the experimental stage. Now, after going through this module, you should be able to explain gene therapy and its approach, discuss about different types of gene therapy, and list various types of vectors which are used for gene therapy, and then discuss the challenges and risks associated with the technique. Basically, many diseases, they involve alterations in the genes leading to abnormal functioning or non-functioning proteins. So, gene therapy works on the premise that diseases can be treated if the gene is corrected and replaced. As you know that many diseases involve alterations in the genes resulting into the formation of non-functional or abnormal functioning proteins. So, gene therapy basically works on the premise that if these genes can be replaced, the protein or the disease can be corrected. So basically, gene therapy is a novel treatment method which utilizes genes as the therapeutic molecules instead of conventional drug molecules. Treatment of these hereditary and acquired genetic defects can be done using this therapy. So first time it was used in 1990 when it was successfully used to treat a genetic disorder known as SCID, that is severe combined immunodeficiency disease, in a 4-year-old girl, Ashanti De Silva. Let's discuss the different strategies of gene therapy. First is gene augmentation therapy. Gene augmentation therapy is used for diseases caused by loss of function of a gene. Therefore, introducing extra copies of the normal gene may increase the amount of normal gene product to a level where the normal phenotype is restored. As a result, GAT is targeted at clinical disorders where pathogenesis is reversible, and it has been particularly applied to autosomal recessive disorders where even the modest expression levels of an introduced gene may make a substantial difference. Second approach is direct cell killing. It's a general approach which is popular in cancer gene therapies. Genes are directed to the target cells and then expressed so as to cause cell killing. Direct cell killing is possible if the inserted genes are expressed to produce a lethal toxin, which will be known as suicide genes, or a gene encoding a prodrug is inserted. So as a result, subsequently, when there is an exposure to drug, the cells will be killed. In addition to direct cell killing, there is another approach which is known as indirect cell killing. It uses immune stimulatory genes such as foreign antigen gene or cytokine genes to provoke or enhance an immune response against the target cell. Third strategy is targeting inhibition of gene expression. This strategy is to block the expression of any diseased gene or a new gene expressing a protein which is harmful for a cell. This is particularly suitable for treating infectious diseases and some cancers. The figure you are looking at the screen shows inhibition of gene expression by insertion of the antisense mRNA coding gene inserted vector or triplex forming oligonucleotides, which is written as TFO, or antisense oligonucleotide written as ODN in the slide, either by forming DNA RNA triplex inside the nucleus or by forming RNA RNA duplex by forming the complementary mRNA strand of the disease protein coding mRNA. This may lead to the blocking of disease causing protein expression. The fourth strategy is targeted gene mutation inhibition. It is used to correct a defective gene to restore its function, which can be done at genetic level by homologous recombination or at mRNA level by using therapeutic ribozymes or therapeutic RNA editing. The figure shows swapping of the abnormal copy of the gene M with the normal copy of the gene, that is X, by homologous recombination. The diseased cells will become normal after the correction of mutation by gene therapy. So let's discuss the process of gene therapy. The first part would be to identify the non-functional gene which causes the specific condition. Second, you have to identify the location of the affected cells in the body, tissue or organ. Third will be to isolate the functional form of the gene. This functional form of the gene is also known as therapeutic gene. And finally, you have to deliver the functional gene into the cells. Depending upon the type of cells in which the therapeutic gene is introduced, gene therapy is of two types: somatic cell gene therapy and germline gene therapy. As is indicated by the name itself, in somatic cell gene therapy, therapeutic gene is introduced in the somatic cells like bone marrow, skin or blood cells, and the effect of these genes is restricted to the patient and is not inheritable to the next generation. At present, all the researches in the area of gene therapy are focused on correcting genetic diseases in somatic cells. Germline gene therapy involves introduction of therapeutic genes into reproductive cells like egg or sperm. Therefore, the effect of this therapy are inheritable. However, due to safety, technical, ethical reasons, germ cell line gene therapy is not attempted at present. Conceptually, the process seems to be very simple, but there are a number of technical problems, and the most critical part is efficient delivery of therapeutic gene inside the cell and make it express. Therefore, specialized vehicle known as vectors are used for delivery of DNA into the cell. The vectors are supposed to ensure the delivery of gene into the target cells and their nucleus, to protect gene from cellular degradation, and also to ensure the transcription of the gene so as to make the functional protein. The figure explains the mode of delivery of gene. So there are basically two modes: ex vivo gene therapy, in which cells are taken from a particular organ of the patient and cultured in the lab. Therapeutic gene is introduced by cloning. Transformed cells are selected, regrown in the cell culture and reintroduced into the patient. Since this technique involves patient's own cells, therefore risk of rejection or other adverse immunological reactions to the transplanted cells are minimized. This is more common method of gene delivery. Another approach is in vivo gene therapy. The process involves direct delivery of the therapeutic gene, that is DNA, into the target cells of a particular tissue of a patient. Therapeutic gene is incorporated in a vector by recombinant DNA technology. Recombinant vector is then introduced into cells of specific target tissue where they express the functional protein. Many tissues like liver, muscle, skin, spleen, lung, brain and blood cells are the potential candidates for this approach. So, you have already seen that efficiency of both ex vivo and in vivo approaches of gene therapy depends upon stable incorporation of the therapeutic gene so that it can be continuously expressed. Therefore, the role of vectors is very important. So, let us look at some of the properties of an ideal vector. First, it should be able to efficiently deliver one or more genes of the size which is needed for the clinical application. Secondly, it should be easily purified in large quantities and available in high concentrations. Third, it should be incapable of autonomous replication of its own DNA inside the cell. Next, it should be very specific for the target cells so as to have minimum side effects. Then it should result into a sustained and stable gene expression, and this expression should be over a long period of time, generally till the life of the patients. Next, it should not induce any immune inflammatory response into the patient, and last but not the least, it should be safe not only to the patients but also to our environment. The figure shows the type of vectors which are commonly used in gene therapy. So there are viral and non-viral vectors. Gene transfer via viral vectors is known as transduction, and via naked plasmids or non-viral vectors is known as transfection. Let's first talk about viral vectors. These are of two types, integrating and non-integrating. Why they have been chosen as vectors? Because they are very efficient in introducing their DNA into cells. Taking advantage of this quality, viruses have been engineered to remove essential genes involved in their assembly, replication, or infection and introduce therapeutic genes instead to be delivered with high efficiency into the target cells. Such vectors lose their ability to reproduce target cells, ensuring safety of the patient. However, these viral vectors can be replicated in specialized cell lines called packaging cell lines, which provide the function of depleted viral genes, allowing production of recombinant viruses in large numbers. Let's see how integrating viral vectors and non-integrating viral vectors differ from each other. Integrating viral vectors like lenti or retroviruses integrate into human genome. As a result, the modifications which are made in this DNA will be passed on to daughter cells derived from these transfected cells. Whereas non-integrating vectors stay in the nucleus without integrating into chromosomal DNA. Human adenovirus, adeno-associated virus, and herpes simplex virus belong to this category. This table shows a comparison of different viral vectors. As you can see that retrovirus has advantage of high transduction rate, as well as adenovirus also shows high transduction rate. In case of lentivirus, immunogenicity is not there and the sustained gene expression is seen, whereas the disadvantage is that there are chances of insertional mutagenesis and transduction is limited only to the dividing cells. Adeno-associated virus show little or no immunogenicity, and transduction can occur in both dividing as well as non-dividing cells. The gene expression is also stable. However, the disadvantage is that they can accommodate only very small size of transgene and the production of high titers is difficult. Let's see what are non-viral vectors. Actually, the non-viral techniques of gene transfer represent a simple and safer alternative to viral vectors. These include naked DNA vector, which are based on the plasmids, the extra chromosomal DNA in bacteria. So, therapeutic genes are inserted into plasmids and recombinant plasmid is then introduced into the target tissue. Injection of naked DNA into tumor can be done by simple needle and syringe. However, this method is not very effective. So another method which is used is the DNA coated with surface of gold particles are air propelled into the epidermis. So basically they are coated with gold pellets on the surface and then injected under pressure into the skin. So this is known as gene gun method. However, it is not applicable to cancer cells. Another non-viral vector is liposomes. You have read about liposomes. You must be knowing about liposomes because these are the organized lipid structures. When membranes are put into aqueous environment, so they form circular organized structures which are known as liposomes or micelles. So the plasmid DNA is basically enclosed into these liposomes, and the complex which is formed by these two components is known as lipoplex. So formation of lipoplex is dependent upon several physical conditions like pH, charge, as well as structural characteristics of the liposomes. These liposomes enclosing DNA, they can be transferred into cells by intravascular, intratracheal, intraperitoneal or intracolonic routes. So basically the liposomes, they are the synthetic vectors which have been designed to deliver the non-viral DNA into the cells without degradation. Let's see what are the advantages of non-viral vectors. First, they are safe because they do not induce any immune response. Second, they can take unlimited amount of transgene. And thirdly, the possibility of repeated administration is there. Limitation, because they cannot produce stable and long-lasting expression, therefore the need for repeated application is there. This table is showing you the comparative analysis of viral vectors and the non-viral vectors. So as you can see in this table that non-viral vectors are very good in taking large amount of DNA. Then they can be induced by ex vivo as well as in vivo methods, and they have very good stability, easy to scale up, no immunological responses, and host immunity does not affect it, and safety is also much better than viral vectors. The only part where they are not better than viral vectors is integration is comparatively poor, then duration of expression is also short. So that is why the need for repeated application is there. The field of gene therapy is relatively new and scientists are facing new problems and challenges every day. Therefore, many risks are associated with this technique, and the utmost concern is safety. First risk associated with gene therapy is lack of long-term expression of therapeutic gene. Now, it's very crucial that the therapeutic gene which is introduced into target cell remains functional and stable for gene therapy to be effective. It has been seen that even integrating viral vectors are not able to associate permanently, and patients will have to undergo multiple rounds of gene therapy. Therefore, moreover, sometimes new gene fails to express or the virus does not produce the desired response. Secondly, there are problems with the immune responses. Vectors are foreign to human bodies, and therefore chances of stimulating the immune response are always there, which might reduce the efficacy of gene therapy. Third problem which is generally associated is there is always a risk from viral vectors because these are although suitably engineered so as not to cause any adverse effect in the patients, number of problems still have been reported. These include toxicity, immune responses, inflammatory responses, gene control, and reaching the target tissue. Moreover, there is always a fear that viral vector might revert to original form once inside a human body. Then comes multigenic disorders. Gene therapy approach is best suited for disorders or conditions which arise from mutation in a single gene. Therefore, some of the most commonly occurring diseases like heart disease, Alzheimer's, arthritis, diabetes, which are due to combined effect of many genes, may be difficult to treat with gene therapy. Target cell is also an issue. Sometimes virus may not target the correct type of cells. So, if DNA is integrated into a wrong place in the genome, say for example tumor suppressing gene, it could lead to the diseases like cancer. Such case was reported in clinical trials of X-linked SCID patients in which therapeutic gene in viral vector was incorrectly incorporated into hematopoietic stem cells, leading to the development of T-cell leukemia in 3 out of 20 patients. Now, gene therapy is a relatively newer line of treatment and involves altered genes and potentially dangerous viruses. Therefore, long-term effects are not extensively studied, and it also raises certain ethical and social concerns. Let's look at those. How can a good or bad use of gene therapy be distinguished? Secondly, who decides what traits are normal and which constitute a disability or a disorder? Will the high cost of gene therapy make it available only to wealthy people? Could the widespread use of gene therapy make society less accepting of the people who are different? And should people be allowed to use this approach of gene therapy to enhance basic human traits such as height, intelligence, or athletic ability? This table is basically showing that initial experiments using gene therapy have been conducted primarily in patients for whom other treatments have failed so that the risks are small. So this table shows the number of diseases like SCID, hemophilia A and B, thalassemia, Duchenne, muscular dystrophy, cystic fibrosis are being targeted for treatment by gene therapy. So students, let's now summarize what we have learned so far in this module. First, gene therapy helps us to treat the genetic disorders that are caused by malfunctioning of the protein. Introduction of one or more genes into the somatic or germ cell lines is done using vectors which could be of viral nature or non-viral nature. These vectors vary in terms of their efficiency of transduction, the size of DNA which they can transduce, the target cells, then immunogenicity and the side effects, as well as the stability of the expression of transduced DNA. So basically the area of gene therapy is still in the development stages and is associated with many risks as well as social and ethical concerns which need to be addressed. Thank you.